Well, the really interesting thing is I teach invertebrate zoology and science fiction writing.
Good evening, everyone at Lollipops and everyone on Facebook Live. Thank you guys so much for joining us tonight. We got some super awesome stuff coming your way. We're going to be talking about sponges. Not SpongeBob was going to be my joke. Hit it too fast. But we're going to be talking about sponges underwater. And we got Dr. Joseph Pawlik here. We'll be starting in just a few minutes, so stay tuned, and then we'll enjoy our evening.
And how many people have said Dr. Joseph Pawlik?
All right. Hey, guys. Oh, almost on. Well, welcome everybody to a wonderful evening. It looks quite beautiful here outside at Lollipops. First, we do want to give a big thank you to our sponsors, including Lollipops, for hosting us here tonight. Thank you, thank you. And to Carib Trans, thank you for your support. All the wonderful sponsors on this beautiful slideshow. Yeah. So first, we actually have an introduction video of See and Learn. So we'll watch that video, and then we'll introduce you to tonight's expert. All right, guys. So welcome to presentation number 3 of See and Learn 2019. Tonight, we have Dr. Joseph Pawlik. So I'll give you guys just a little bit of background on him. Joseph Pawlik received his bachelor's in 1982 from the University of Minnesota Twin Cities and his PhD in marine biology in 1988 from Scripps Institute of Oceanography, UC San Diego. He joined the Department of Biology and Marine Biology at University North Carolina, Wilmington in 1991, where he teaches an undergraduate course in invertebrate zoology and biodiversity, teaches Introduction to Science as a Profession to graduate students, and directs a research program involving undergraduate, master's, and PhD students. Joe worked at the U.S. National Science Foundation as a program officer in the Biological Oceanography program for 2 years beginning in 2003. Tonight, he will be presenting on the vicious circle hypothesis and the state of the Caribbean coral reefs. Please welcome Joseph Pawlik.
Well, I want to begin by thanking Tiffany and Emily and most of all Lynn for keeping this program going. The See and Learn program is, I don't know of another thing like this. It's absolutely amazing. And I was here 5 years ago, as I'll mention in a few minutes, but it's astonishing that you all are still, keeping this going, and there's so much support from the community. So I want to start by showing you a video that I took this morning at Twilight Zone, and it's a little light. But you can see that the reefs here around Saba are dominated by sponges. No question that this is the highest cover on the bottom, so the amount of area that's covered by the reef is mostly sponge. And when you consider how robust these sponges are, how large they are, there's no question that it's the greatest biomass on the reef. And I would show you the few corals that are still on these reefs. There are some here. But really, the cover is mostly macroalgae and sponge. And then, of course, my favorite sponges are represented, the giant barrel sponge, if you can see that in this video. So that was just from this morning. No question that these are really the big players on reefs here. I've been working on sponges on Caribbean reefs for 30 years. And most scientists, when they start a project, they usually reach an end point after about 5 years, and then they kind of have to switch gears and move to something else. And I've been really lucky to have picked a project that's just gotten more interesting and developed a layer upon layer of more interesting science with essentially a continuous narrative as far as the story goes. And today I'm going to tell you about Caribbean reefs in general, and a particular hypothesis that we've come up with that references the important role that sponges play on Caribbean reefs. But as I mentioned, I was here 5 years ago, and I told a different story. I told a story that actually segues into the story I'm about to tell you. And in order to make all of this fit together, I'm going to repeat what was essentially the most important thing, the takeaway message from the talk I gave 5 years ago. And that is that the sponges that are on Caribbean reefs, some of them have chemical defenses. And what I mean by that is their tissues have unusual compounds in them, chemical compounds, that make their tissues unpalatable or taste bad to potential predators, okay? And also on these reefs, there are sponges that are palatable, that the sponge-eating organisms actually prefer to eat. Those don't have nasty chemicals in them, but as a trade-off, they grow faster. So they grow faster, they reproduce faster, and actually they heal themselves faster. On Ceba's reefs, you have both of these categories, but the palatable ones are terribly grazed. You can find them on all of these reefs, but all of the big sponges that you see are the ones that are chemically defended. You also have preferred species. Those get completely wiped off the reef. Not only are they lacking in chemical defenses, but they also grow slower. And so when sponge predators are around, they totally eliminate that category of sponge. Now, there are 700 species of sponges on Caribbean reefs, 700, of which about 50 are common, of which about 20 in any given site are common. And for example, in Ceiba, there are really like maybe 10 that you see pretty much everywhere. They're the most abundant ones. And so it's fairly easy to study the ecology of the system and not have to reference specifically the species. You can actually reference them by category. So keep that kind of in the back of your mind, because I'm going to switch gears here and talk more about why coral reefs are dying. All right? You've all probably heard that the Great Barrier Reef bleached twice in the last 3 years. Devastating impacts. And these are pictures of a bleaching event on the Great Barrier Reef. And what happens generally is the corals bleach. They lose their symbiotic plants that live inside their tissues. And then the surfaces of those corals are completely covered by algae. An interesting thing, though, about this process is that on Indo-Pacific reefs, for the most part, this process has generally been one of recovery. So you get an event where you've got like maybe 60% cover of coral. Can you see that? Okay, good. Just barely, but you can see it. It'll be getting better, I'm sure. Some 60% cover, a bleaching event occurs. It's all, the corals are down to 10%, but, as quickly as 2 years later, baby corals recruit, and here we are in 2014 from 2006, so less than 10 years later, and you have the same coral cover that you had when you started. So what this is called is resilient, a resilient reef. And Indo-Pacific reefs, for the most part, have been resilient. Interestingly, in the Caribbean, this has not been the case. So in the Caribbean, we had incredible reefs right up until the late 1970s, into the 1980s. And actually there was a pathogen introduced that killed our branching corals primarily. And the result was devastation and no recovery. So Caribbean reefs are not resilient. And this has been a subject that coral reef scientists have been puzzling over since the 1980s. Why is it that Caribbean reefs are less resilient than Indo-Pacific reefs? And what I'm here to tell you is that maybe sponges are part of the reason for that. And that's a hypothesis that we've recently come up with. So just to recap, you already saw what Ceba's reefs look like, right? But Ceiba really isn't a good place to try to study Caribbean reefs because Ceiba is a volcano, and basically it has coral growing on the side of the volcano. The real reefs that are similar to reefs in other parts of the world are the ones that grow along the Mexican and Belizean coast, where we actually have a barrier reef, and similarly on many islands that are in the Bahamas, where you essentially have a thick reef made up of limestone skeleton of thousands of years of coral growth. That's really more sort of what the kinds of reefs that are similar to Indo-Pacific reefs are like. But despite that, the difference between Caribbean and Indo-Pacific reefs is that the cover of sponges is much higher than it is in the Indo-Pacific. So in fact, now the cover of sponge, so surface area that's covered by sponges, is about the same as what corals are, and it's really small on Indo-Pacific reefs. You're lucky to find sponges at all. Moreover, the morphology of sponges on Caribbean reefs is very different. You get all manner of barrels, tubes, branches. It's very variable, but in the Indo-Pacific, they actually look like plants, and they behave like plants. They get most of their nutrition from cyanobacteria that live in their tissue. So they actually behave like corals, because if you know anything about corals, you know they feed themselves by using plants that live in their tissues. So actually the sponges do the same thing there, and they don't do that in the Caribbean. So here's the average sort of Caribbean reef, and this is off Roatan, and you'll see lots of sponges, okay? Lots of barrel sponges, lots of tube-forming sponges. You see some coral interspersed in here, and you see a lot of algae as well. And I don't expect you to see that level of, but trust me. This is very different than the sorts of reefs you see in the Indo-Pacific. So here, nice tube sponges, big barrel sponges. Here's another kind of tube sponge. These are all the main species that are represented. This is what an Indo-Pacific reef looks like. 100% coral cover. Lots of different species of corals. No sponges. No algae. Really hard to find algae or sponges. So this is a very different kind of reef. This is a Red Sea reef off of the Egyptian coast. These are some of the best looking reefs still left. For whatever reason, they're resistant to climate change, temperature problems. So very different systems. And most people talk about coral reefs in general, but in fact, coral reefs are very different depending on where in the world you are. Well, we've been studying sponges on Caribbean reefs for since the 90s, and we've actually been monitoring populations of sponges since 1997. And we really focused on the giant barrel sponge, which is very common here. It gets very large. There are plots that we have continuously monitored so that we monitor everything from the little babies. So this is a person's thumb here, and that's a baby giant barrel sponge, the same size as the thumb. We drive stakes next to all the babies that we find, and we essentially take pictures of them every year to monitor their growth. That's how we figure out how fast they grow and what the relative amount of sponge on the reef is. And this graph shows you increasing abundance of sponges on our continuously monitored time series plots in Florida. And the point here is that the abundance of just barrel sponges has increased 122% between 2000 and 2012. Their volume has increased by 40%, and there are now 2 liters of sponge per meter square of reef. So that's a full quart volume of just that species on every meter square of Florida reef. That's pretty substantial, and you have at least that out here, if not more. So these sponges are abundant, and they're increasing in their abundance. That's the first point. The second thing about them, and the thing that people don't realize, is how much water they process. So this is a barrel sponge that we've added fluorescein to the base, and it's pumping out the fluorescein through the top of the sponge. That's the direction of water flow in the sponge. And I'll be doing this demo tomorrow on a dive, so you'll be able to see what this looks like. And the thing about sponges is you see them, but you think they're just sitting there, just like the corals and the algae. But they're not. They're doing something very important. They're actually processing the water to the point they can actually turn the water column over. If you had a static water column above that sponge, it would turn that entire volume over in as little as 2.3 days. So these are major processors of water. And even scientists don't get this. That's a major sort of effect. And as they're doing that, they're changing the chemistry and the biology of the water that they're processing. The primary reason they do this, the reason they pump water through themselves, is to eat. And the first thing that they eat are particles. So we call this particulate organic carbon, and those particles are both live and dead. The live particles are mostly microbes, and those microbes are either bacteria or tiny little plants that are, some are bacteria, some are not. The other thing they eat, interestingly enough, and the thing that many people don't realize, is they also eat dissolved organic carbon. And the DOC, or dissolved organic carbon, is made up of two categories. It's actually made up of a spectrum of categories. There's labile organic dissolved organic carbon, and there's refractory dissolved organic carbon. And the easiest way for me to explain that is when you get up in the morning and you have a cup of coffee, right? You pour the coffee in the cup, and you add 2 scoops of sugar, right? And you stir it, right? The labile dissolved organic carbon is the sugar. The refractory dissolved organic carbon is the brown color, right? The brown color is actually tannins. It passes right through you. You don't absorb it, but you do absorb the sugar. So, really easy to understand. Labile and refractory dissolved organic carbon. So that's actually in water. In fact, dissolved organic carbon is the largest carbon pool or reservoir in the biosphere that's known, because the ocean is, 70% of the cover of the planet, and there's a lot of volume there, and there's a huge amount of dissolved organic carbon in that water. So they're actually eating stuff that's totally dissolved. All right, you with me so far? Great. All right, well, we started studying what sponges eat, and we used the barrel sponge as our sort of model species because it's really easy to study the water that's on the outside before it moves through the sponge body and the water that's in the center of that big opening, because all of that water has been processed by the sponge. So all you have to do is go sample the water and you can figure out what the sponge has done to the water. And so one of the things we do is we sample the water, and we use this expensive machine called a flow cytometer that can actually tell you what the particles are in the water. And so these are all the bacteria, and each one of these dots is an individual bacterium. These are all the photosynthetic phytoplankton, another kind of photosynthetic phytoplankton, and these are eukaryotes that are very, very small that actually feed on these guys. Once the water is passed through the sponge, these all disappear. The sponge eats them. It eats most of these as well. You see how this is really dense? That red means lots and lots of them. They vanish. So the sponge basically strips those particles out of the water. And these are incredibly small particles. That was pretty interesting. And remember, these cells actually are like meat. This is the meat that the sponge is eating. They're also eating dissolved organic carbon. Those are particulates, right? But they also eat the dissolved organic carbon. So here are my students busily sucking the water from the inside of the sponge. That's the water that's already been processed by the sponge wall. And here they're sucking the water on the outside. And what they discovered is if you sum up all the food that's in the water, you have very little that's actually those bacteria that I just showed you. There's a much larger group that's actually just dead particles. That's called detritus. And then this huge brown band is all the dissolved organic carbon. That's the largest source of potential food that's in the water as it's going through the sponge. When it comes out of the sponge, the water that's in here, they've eaten most of the live particulate organic carbon, so the meat. They've eaten most of the dirt. They love the dirt. Strangely enough, it turns out the dirt is covered with bacteria, so it's just like eating the meat. And they've eaten a lot of the DOC, but only a certain amount, because some of it is refractory, and it goes right through them potentially, and some of it is the labile kind, like the sugar in your coffee, so that they eat most of it, right? But when you sum it all up, the interesting thing, 90% of their diet is not the living meat. it's 70% the dissolved organic carbon, 20% is the detritus, and only 10% are the particles. And most zoologists just assume that sponges only eat living bacteria, when in fact 90% of their diet is dead stuff, and 70% of it is dissolved. So, sorry? Go ahead, ask the question.
So they're cleaning the water?
They are cleaning the water, right? But as I'm going to tell you, when they're eating that live POC and digesting those particles, what do when you eat food? What's the end product of that process? They pee, just like we do. So that water also has their pee in it, because they're breaking down those live particles. I'll get to that, though. All right, so at the same time we're doing all of this, a Dutch group actually working in Curacao came up with a hypothesis that we ended up testing for emergent sponges. They did this on cryptic sponges, which live in the cracks and crevices in the reef. And they called this the sponge loop because it's similar to a hypothesis that's been advanced for what happens in the water column of the world's ocean. And the idea here is that seaweeds and corals are giving off sugar, like the sugar in your coffee. And that's DOC, dissolved organic carbon, that's being eaten by sponges, but then the sponges produce detritus. You don't have to be too mindful of this part of the hypothesis. It turns out that we've disproved it for large sponges. But the really important thing here is that subsequent studies by many groups have shown that when seaweeds are exposed to light, only half of the sugar they produce do they use to make more seaweed, do they use to grow. Half of it they just give off as sugar into the water. So that sugar is then subject to being eaten by microbes, but if the sponges can get it first, they can eat it. So this is a potential major source of food, labile dissolved organic carbon, for the sponges. And this is really interesting. This is the first part of what we consider the vicious circle. Okay, so part one, I'm gonna do a summary slide so that you can all kind of catch up here in a second. The last piece of the puzzle I want to talk about is the P part. Okay, so when people who were doing geochemistry, so studying nitrogen cycles in the sea, put their little probes over sponges, they came to the conclusion that in fact sponges are producing huge amounts of fertilizer. And that's essentially nitrogenous wastes, essentially P. Ammonia, nitrite, and nitrate are the forms of P that sponges make. Do any of you know what the form of nitrogen that we all make is? Do you know what? Urea. We make urea because we can't deal with these more simple nitrogenous wastes. But this is a huge amount of fertilizer that the sponges are essentially producing as P, and it's more complicated than that because it actually turns out that there are microbes that live in their tissue that are also doing complicated conversions of the nitrogen that's present in the water. So, but for the purposes of this talk, the thing to remember is that sponges make a lot of P. And that's a part and parcel with eating a lot of these particles, right? That's just going to happen. So here's the summary slide for this talk up to this point. Sponges in the Caribbean, they're abundant, they're increasing in abundance, they turn over a lot of water, they eat mostly dissolved organic carbon, sugar in the water, and they excrete a lot of fertilizer, right? So those are the important things. And, you know, the thing in science is generally studies are done that come up with parts of a puzzle, and what it takes is somebody to sort of put all the pieces together, and that usually causes the science to advance. This happens frequently. And so, before I get to putting all that together, I want to talk about one other interesting fact, and that is the geography of the Caribbean Sea, because the Caribbean is different than the Indo-Pacific, because it's like a big mixing bowl. If you look at the surface currents in the Caribbean, and you kind of squint, you see that in fact it's like the arrows are all kind of going in a circle. I realize they seem to be going the opposite direction here, but there are currents that actually run on this side of Cuba as well. And the way they really know this is the physical oceanographers release these tiny little cups that float and that have transponders that talk to satellites, and you get the spaghetti. But you notice how the spaghetti is very circular. There are lots of circles here. From the biological side of things, we know that the Caribbean is a big mixing bowl because every time there's a disaster, a biological disaster in the Caribbean, it spreads around the entire Caribbean really rapidly. It happened in the 1980s when both coral diseases were introduced to the Caribbean and when a very terrible die-off occurred of the long-spine sea urchin. This happened in 1983, and it happened in 6 months. Basically, all the urchins in the entire Caribbean, they're used to, any of you go diving in the Caribbean in the 80s? He asked, ridiculous. Some of you, hey, you couldn't, go a meter without seeing 2 or 3 giant long-spine sea urchins. They were everywhere. And then all of a sudden, in 1 year, they all died. And it's because a disease was introduced, probably from the Pacific, that killed all of them. What's the most recent example of the mixing bowl effect? Lionfish, right? You introduce them one place, the next thing you know, they're everywhere. So we know that this is true. But actually, the physical oceanographers have known this for a long time because there's a very specific signature in the water of the Caribbean. And the reason it's golden. So if you do a fluorescence on Caribbean surface water, it always comes out golden. And the reason for that is there's a lot of river input into the Caribbean Sea. From the Mississippi, the Cartagena River, the Orinoco, which comes in just south of Tobago and Trinidad. And then it turns out that the Amazon River, for 6 months out of the year, blows directly into the surface of the Caribbean. So essentially that fresh water sits on the top of the Atlantic, and the wind blows it right into the Caribbean. So the interesting thing about that river water is it's full of dissolved organic carbon. And we think that this may explain why the sponges in the Caribbean are so different than the sponges in Indo-Pacific reefs, that they have essentially evolved to take advantage of some of the more refractory, some of the brown water that's in your coffee that's also in this river water that the microbes can't seem to take advantage of. All right? So let's put all of the pieces of the puzzle together, and we get something called the vicious circle. And this is the complicated version of the vicious circle. And so what's happening in the Caribbean is we have similar abundances of sponges, seaweeds, and corals, and there's this feedback between the 3 groups in terms of dissolved organic carbon, sugar in the water, and nutrients, fertilizer. And then there's the input of rivers into the system. There's the pee that comes from fishes, there's the pee that comes from sponges, and there's the interaction with microbes. If you want to simplify this, it looks like this. Basically, the seaweeds make dissolved organic carbon, the sugar. The sponges eat the sugar. Remember, that's 70% of their diet. They pee the fertilizer, and they fertilize the plants. They take over the real estate. The corals die, leaving them more real estate, and they hold more and more real estate as they feedback loop between food and fertilizer. Understood? Any questions? Question? Oh, I'm glad you're going to ask that because it comes next. He asked, why hasn't this happened in the past? I have a good explanation, okay? But first, let me show you why this doesn't happen in the Indo-Pacific, right? Go ahead.
Do the sponges also eat the eggs or whatever, however the coral reproduces?
Those actually are particles that are too large for sponges to eat. Other things will eat them, but sponges are actually adapted to eat fairly tiny particles. If they fall directly on the surface, they can phagocytose them, but that almost never happens. It's a very rare occurrence. So why doesn't the vicious circle, first of all, happen in the Indo-Pacific? The reason there aren't any sponges. There's no river input. And so you can't get the feedback. There are very few seaweeds. So up to this point, you can't get the feedback loop between sponges and the seaweeds. The seaweeds don't generate enough dissolved organic carbon. The sponges can't produce the fertilizer. Question.
Does seaweed include algae?
It is all algae. Yeah, yep. I tend to always use the simpler word just because it's easier, right? Okay, so that's why it's not happening in the Indo-Pacific.
Why isn't there seaweed and algae in the Indo-Pacific?
Because the corals, for the most part, do pretty well, and they're holding that territory. And as long as there isn't the levels of DOC that help the sponges grow, the only way the sponges survive is by making their own food by using plants. So they grow like plants. And they always have grown like plants. In the past, they have as well. Okay? All right. So the gentleman asked, why don't you have a vicious circle always? Why hasn't this been a historically problematical situation? Why is it only recently that the vicious circle has become a problem? Well, now I have to go back to what I told you 5 years ago, which, remember, there are these fish that eat sponges. They will eat the palatable and the preferred species off the reef if they're there in abundance, but they really won't touch the defended sponges because the defended sponges have chemical defenses. They taste bad, and so they just tend not to eat them. The most common predators of sponges on the reefs that we see here or anywhere else in the Caribbean are all the angelfishes. In fact, these guys are major spongivores. This one is a very specific sponge feeder, the rock beauty. The parrotfish also are major spongivores. They love sponges, and they actually have their own separate set of preferred species. But the other major sponge feeder is something you rarely see, or until recently rarely see, and those are hawksbill turtles. Now the answer to your question, in short, is that we used to have a lot more of these. And the reason we know that is because when Europeans first invaded the New World, they were very conscious of making it back home. They were very worried about making it back home once they got here. And one of the things the captains did and spent a lot of time doing is writing extensive logs that explained how to get to the New World, but also how to find fresh water and how to find food. And one of the major sources of food for people who lived on ships was turtle meat. And more importantly, the eggs of turtles, because they kept. You could keep them in the hold for long periods of time. They were a high protein, nutritious food. So the captains of these ships kept logs, and the logs were hugely valuable. In fact, they used to steal one another's logs, and they would sell captains' logs for huge amounts of money because they basically revealed all sorts of secrets about where's the food, where's the island, where's the freshwater source. So there's a group of people, scientists called historical ecologists, who pore over captains' logs in the British Museum to learn things about what these ecosystems were like at the time that these captains were exploring the New World back in the 15, 16, and 1700s. And what they determined by back calculation, so using the number of nesting beaches and the number of bumps and between the number of times that the ships were striking turtles, their estimate is that hawksbill turtles in the 1680s were about 11 million turtles in the Caribbean. By 1974, there were 27,000. So that means that for every hawksbill turtle you see now, there were 400 hawksbill turtles. Imagine that. And that's also true for green turtles, which are more algae eaters. But these hawksbill turtles are sponge eaters. Now, interestingly, the reason that they captured hawksbills back then was not for their meat. They would eat their eggs, but they wouldn't eat their meat. And the reason they wouldn't eat their meat is because it would make them vomit excessively. right? So they would collect hawksbills. You know what they used hawksbill shells for? Combs, all sorts of corset stays. So they were very valuable commodities, and they brought them back, but they wouldn't eat the flesh. By the 1900s, the flesh was edible, and by the 1974, totally edible. Why? Because at this time, when there were 11 million of them, these hawksbills were eating all the sponges. By the time there were only 27,000 of them, they were eating only the tasty sponges, and this is their favorite. So what this means is that back when there were 11 million turtles in the Caribbean eating sponges, they ate the sponges completely off the reef, and that's what prevented the vicious circle. They essentially were consuming, as major predators, all of the sponges. By the time we reach the modern day, and interestingly, there were still over 1 million of them by 1900, and only then was the meat essentially sufficiently less toxic that you could eat it. So it's been relatively recently that turtles haven't been having a major impact on the sponge fauna. But by the 1970s, they're having virtually no impact. So
the turtles were chemically defended by eating the sponges?
Chemically defended to humans by eating the sponges. And the thing about reptiles is, just like you and I mean, if you're starving, you'll eat all sorts of things that you wouldn't eat if you had your preference. I mean, are you going to eat a steak or a rice cake, right? So it's the same thing for them. They're going to eat their preferred foods, and it turns out their preferred foods are the palatable species of sponges. Okay, so pre-Columbus reefs had high coral cover, low seaweed and sponge cover, because they had a high abundance of turtles, fishes, and urchin consumers, urchins being the things that were devastated in the 80s. At that time, river input was from pristine wetlands. So all of those rivers had really high quality water flowing into the Caribbean, but there was likely a high amount of refractory dissolved organic carbon. Where were the sponges? They were living in the interstices of the reef. The cool thing about sponges is they can just hide. They can retreat to the places where the spongivorous fishes and turtles can't hurt them. Between the 1700s and the 1900s, we had overfishing of turtles and fishes, and a huge drop in the numbers of hawksbill turtles. Nevertheless, the coral was still there, holding its real estate. Then all of a sudden in the 1980s, we transport pathogens, which kill the corals and the urchins, a 1-2 punch from which Caribbean reefs will probably never recover in our lifetimes. Low coral cover suddenly, freeing up all this real estate, high seaweed and growing sponge cover. Seaweeds grow very rapidly, sponges much less so the sponges were slow to take over, but the vicious circle essentially started. Low abundance of seaweed and sponge consumers, so they couldn't control the populations of sponges or seaweeds, and by now river input is from impacted watersheds. So potentially we've got water that's coming in that has higher levels of refractory organic carbon and potentially higher nutrients. The present, very low coral cover, high seaweed and growing sponge cover, the vicious circle is established. Recovering consumers cannot keep up. And river input from impacted watersheds. So, a very sad story. No, that's actually a whole nother story. But basically, the coral has its own microbiome, just like we all do. And that microbiome is subject to changes that enhance pathogenicity. So we're now thinking that a lot of coral diseases are less on account of a pathogenic effect, but rather because their microbiome has been transformed and they become sick from it. They're stressed. Okay, so this sort of shows you what I've been talking about kind of in in 2 cartoons. Back pre-Columbian Caribbean reefs, lots of turtles and fishes busily grazing down the sponges, forcing them into the interstices of the reef. They're all there, but they're hiding, right? And as soon as they grow out, they get munched down because these are hungry turtles eating those sponges as quickly. And remember, for every one that's there now, 400 on this side, based on the estimates that the historical ecologists have come up with. So, lots of beautiful sponges, but not the way it was back in the day. Now, if there's any glimmer of hope in this story, it's this. We have increasingly been bringing turtles back, right? We've been very aggressively protecting their nests. We protect them in the water, and you're not allowed to take turtles anymore in most places in the Caribbean. And so interesting, what we've seen is that the numbers of turtles are rising, and not slowly, actually quite dramatically. In the 90s in Florida, when we would go diving, we were lucky to see a hawksbill once in 2 weeks. And now we see several on every dive. They're all juveniles, and they're coming back, you know, in large numbers. And if you go to most places, you're seeing those turtles busily scarfing down the palatable sponges that they love the most. And now you see, this is Geodia neptuni, Neptune sponge. It's hard to find a Neptune sponge that doesn't have turtle bites in it. Once the turtles open these up, the spongivorous fishes come and gobble up their soft inner sections, because it turns out this particular sponge has a really hard crust. But the turtles are starting to have an impact, first on the most palatable species. So that's kind of a positive thing. I'm almost done. Yeah, so that's the glimmer of hope in all of this. So in closing, I want to thank all sorts of people. Obviously, I didn't do any of this alone. This is, you know, years of work with many, many students and many collaborators, and generous support from the National Science Foundation, the use of all sorts of facilities that have been made available to me, including the Aquarius habitat that's at 50 feet off of Florida, the Smithsonian Institution, but by far my biggest funder has been the U.S. National Science Foundation. And with that, I thank you for your kind attention. Happy to answer any questions you may have.
For the predator fish and the hawksbill turtles, has eating the sponges over time affected their physiology?
The question is eating sponges something that would affect the physiology of the predators that eat them, right? And the interesting thing there is for the spongivorous fishes, they have a gut that's adapted more to digest the skeleton of the sponge. And other fishes, even when the sponge is palatable, they won't eat it because they can't get enough nutrition out of it in the time it passes through their gut. And so, yes, there is a physiological change in their gut that makes them better able to get nutrition from sponge tissue. For turtles, the really interesting thing about hawksbill turtles, because you know there are hawksbills and greens and loggerheads and Ridleys, Kemp Ridley turtles. For whatever reason, just the hawksbills eat sponges. And they seem to have, similarly, a different kind of gut. And one of the really interesting things about their guts is their gut is able to withstand the passage of lots of tiny glass shards through their gut, because most of these sponges have tiny little glass needles in their tissue. And this doesn't seem to be a problem for any of the things that eat sponges. It would be like eating fiberglass insulation, right? You just can't imagine. And when you dissect a hawksbill turtle, it turns out that these things perforate their gut frequently, but it doesn't seem to affect them. But fishes and reptiles are well known to be able to eat all sorts of strange things and not have any real negative consequences. For whatever reason, the green turtles have a different gut that largely digests plant material. So that's what they've specialized in.
Yeah, so with the essence of the depths of the reefs and whatnot. So we dive on a lot of shallow reefs. Often, some tech diving friends and people in industry, they dive a lot deeper in the sponge zone itself, since I guess some corals can't grow there.
Right.
So can you comment on that zone and maybe its effect on the shallow reefs, whether it pumps water up, or if that's always been sponge territory throughout all the time, or its effects?
Great question, Andrew. So the question is, there's a deeper area in the mesophotic, which is a deep zone, where there are lots of sponges, and there are no corals. And the reason there are no corals is because the light levels are so low that the corals can't make food, and so their abundance drops. And we just published a paper on this, actually, because we surveyed the mesophotic reefs off of Puerto Rico. And what we discovered was you do get an increase in abundance of sponge with depth, probably for competitive reasons, because the corals essentially give up their space as the light levels decrease. The sponges get larger, and we think the other reason the sponges get larger with depth is that, some of the giant barrel sponges we study are thousands of years old. And one of the things we thought was weird was that off of Florida, you never find large sponges. And similarly, there are places where they reach a certain size, and that's about as big as they get. It turns out that if you're close to continental areas where there's sediment, it only takes one major hurricane event to push the sediment into the water to smother the sponges. And if you have one of those events every 300 to 500 years, you kill all the largest sponges, and they have to recruit from the smaller ones. You go to the off islands, like Saba, or better, the off islands of the Bahamas, which are way the heck out in the middle of the Atlantic, they never have this problem. So you get these ginormous barrel sponges that get really large. In the mesophotic zone, similarly, you don't have these problems. There are no terrestrial influences of those sponges. So they're probably really old and have never been molested by any of the things that might bother over long spans of time the sponges that are in shallower water. Moreover, there seems to be an increase in the amount of the particulate food in a band in deeper water. So the Prochlorococcus, these are actually cyanobacteria. They're blue-green algae. They grow at depth, and they use low light levels. And the sponges are taking advantage of that additional meat that they don't have in the surface water, right? They'd rather eat meat than sugar. And they probably grow faster because of it. And because that band is in deeper water, that potentially enhances their growth as well.
Follow up real quick. Is that global, or is that our regional, domestic?
Well, there's some discussion about how globally that's true. But the other thing that this gets complicated, what we've found is they're also relying on the sugar from algae. And even at great depths, you have coral and algae on the walls that are producing those sugar. When the coral and algae drops out, when you get too deep, the sponges drop out too. But you're also below the level of those cyanobacteria too, because there's no light down there. So there's a link between even low levels of light and the abundance of those sponges. Then all of a sudden you get glass sponges. Glass sponges don't take up DOC, turns out. And they're really slow growing, 500 feet. Yeah.
You mentioned the 27,000 is the number of hawksbills, but that was 40 years ago?
1974, yeah.
Okay, we're even longer. Do you know what the number is today?
I don't, and I looked in that paper to see if they had a subsequent estimate, and they didn't. I bet if you go online, you can get estimates. I should have done that myself, but they're always updating Caribbean populations, so I'm sure it's double that at least. So that would be my guess.
So I got two questions, but if that's okay. But the first one is, so the problem is basically fertilizer, right? For the corals.
Yeah. So the first question is, the problem is basically fertilizer. And interestingly, there are some reef biologists that swear the reason our coral reefs are dying is because of human pollution, right? And that's very common. You hear it all the time. And my response is no, it's not, because you can go to reefs far away from humans and they look just as bad as the reefs that are close to humans. And that, to me, totally disproves that, because there's no anthropogenic source of fertilizer. Where's the fertilizer coming from? The sponges. It's a natural source of fertilizer. It's the vicious circle, right? So yes, that is part of the problem, but you can't get the fertilizer unless you feed the sponges, so you got to have the feedback loop. So what's your second question?
My second question is, I'm actually finishing up my master here in Alceba, and I'm doing research on the artificial reefs that are around the island.
Right.
And right now in our research question, we're mainly focused on the macroalgae that are on these reefs. Do you think it would be very interesting to include the sponges in our analysis or not? Because they are, of course, close to other reefs where there's giant sponges.
Right.
So for the bentals cover.
So he's studying the macroalgae on artificial reefs here in Seba. Where are these?
They're around in front of the quarry.
Okay, in front of the quarry, and they're close to shore, or they're
pretty decent.
Yeah, and they're what? They're made out of concrete, or
yeah.
Okay.
It's the reef balls.
Reef balls.
Yeah,
okay.
We're trying to fundamentally discover whether these are important in different types of shapes.
And you're planting corals on them as well? No, you're just watching them naturally aggregate organisms, right? Yeah. Yeah, I think it would be very interesting to see. I would definitely count the sponges, and as cover, it would be interesting to see whether you see an increase in algal abundance as the sponge abundance increases. So, whether there's a gradual algal increase and then suddenly an abrupt increase that's correlated with sponge abundance as well. The thing with sponges is that they're slow to recruit. And we study that on artificial wrecks. So we have used in two places. There's a wreck off of Cayman Brack called the Tibbets, where we know exactly the year the wreck went down, and we can figure out how quickly the sponges grew because we know they could not have been there before the time that the ship was wrecked. And the other wreck that we've studied extensively is the Spiegel Grove off of Key Largo, because we know exactly when it went down as well, and we've learned a lot about sponge growth from that. The problem on the wrecks is that very often algae doesn't grow as well as they would on reef balls. So that's a potentially interesting. Where are you getting your degree from the Netherlands?
Yeah, from Groningen, right?
Okay.
I'm doing this for Wageningen.
Oh, say that again?
I'm doing this for Wageningen, but I'm from Groningen.
Okay, so Wageningen is where you're from, and who's your advisor? Okay. Say the last name?
Dolphy de Brood.
De Brood.
Yeah.
Yes, I do. Yeah. Yeah, I have a hard time understanding. It took me a long time to say De Hui. So and everyone says De Googe. So, yeah. Yeah, okay. Great. That's terrific.
When sponges process to the water, do they heat up the water as it's going through?
No, not that we know of.
When I put my hand inside a barrel sponge, it seems as though the water is marginally warmer inside the barrel sponge than outside, and I'm wondering if that may have an impact on the ambient water temperature.
We've never tested that, but at some level, cellular processing should produce heat. So that's an interesting question. Yeah, I don't know. Yeah, absolutely. It would be infinitesimally small, given the rate at which I can't imagine you could feel it. So I would.
I mean, it could be just in my
I
head,
suspect that it's in your head.
But I do know that if you drink it, if you drink a cold glass of water, when you pee it out, pee is warmer than the water that's
This is true. This is true. You, however, are a warm-blooded organism. Yeah, you're maintaining a temperature. The sponges are not. And they're actually processing water so quickly that even if there was a slight cellular effect of temperature, it should be measurable, but it shouldn't be something you should feel. Interesting question. I've never had that question before. Congratulations. Go ahead, Sabrina.
The vicious cycle.
Circle.
Circle, sorry.
No, cycle is what I wanted to use, but decided not to. It's a long story. Okay.
All right, skipping that. I'm thinking about one that wouldn't be vicious, which would be birth and death, essentially. And you mentioned the age of one of the sponges earlier. So I'm wondering how they reproduce and also how long their typical lifespan is, and is it represented by their size?
Great questions, all of which we've studied. So Sabrina's question is, what do we know about how sponges reproduce, and what do we know about their age relative to their size? Is that a fair estimate of? So there's, as I said, there are 700 species of sponges in the Caribbean alone. There are probably 20,000 worldwide, and that's probably a low estimate because they tend to have cryptic speciation, which is another question. But the interesting thing is that for every one of those species within families, you get a lot of diversity in the way they have evolved to do what they do. And so all of the branching sponges, and for example, reproduce asexually. They will just break up, and when the little pieces land on the bottom, they reattach and start growing. The giant barrel sponge, however, only reproduces sexually, and they are separate sex, just like we are. So they're male and female sponges, and if you're lucky enough to come onto the reef when they're reproducing, the males smoke, so they produce sperm that comes up out of that opening in this amazing white column, and the females make flocculent egg masses that bubble over like soap suds out of a tub onto the reef around them. And the sperm find the eggs, and little planula, like worm-like larvae hatch out of the eggs, settle on the bottom, and grow up into new sponges. Now, sponges don't have annual rings like trees, nor do they have bones that add annual amounts of mineral like the ears of fishes. So the only way we've been able to age sponges is to take pictures of them over time. And if you take pictures in a way that you can estimate the volume relatively accurately, you can figure out what the change was for a given period of time. And then you use something called growth modeling to figure out how old they are. And based on those growth models, a sponge that's about this big, so the size of maybe a 50-gallon drum, is about 200 years old. And the largest sponge that we have a picture of from off the coast of Curacao, we estimated at about 2,000 years old. Unfortunately, when that sponge was photographed, it had a pathogenic condition and it subsequently died. So it's not with us anymore. But I have wonderful video of sponges in the Bahamas that are easily in the thousands of years old, based on our best estimates. Now, it turns out that the more we've studied barrel sponges, the more we realize that especially barrel sponges are hyper-variable to the point where you can watch one individual and it doesn't do anything over time. It never grows, it never changes. Another one will be growing year to year. And only recently have we discovered, using molecular phylogenetics, so essentially you clone the genes of the sponges and you look at differences in their genes, we have discovered there are 3 separate, reproductively isolated species of giant barrel sponges here. And even more fascinating, so basically they look alike, they do all the same things, but they do not interbreed. They have been distinctly different from one another, we have discovered, for at least, it's got to be at least 6 million years, because the diversity within the Caribbean is greater than the diversity in the Indo-Pacific, and the Indo-Pacific are more closely related to one of the groups in the Atlantic, which blows the minds of most people who study how species form. Because basically what that means is barrel sponges speciated into separate, reproductively isolated units before the Pacific and Atlantic oceans separated from one another. And they have been distinct ever since. And this is real new data. So, also from Dutch researchers, Thomas Swijerts from Nicole de Vugd's lab. De Vugd, am I saying that right? De Vugd. Sorry, I don't have enough phlegm.
That's interesting. So with fossil records and the lifespan of life on Earth here, sponges, I guess, are one of the more ancient animals here. And due to all the different extinction events, I guess, 5 or 6 of them throughout the time here, have there been ages of sponges, like an era of sponge of oceans, and now corals have taken over, but now are we going to retrograde back to a sponge ocean, potentially? With what would be your predictions, or if there's any indication in the past that corals weren't the dominant force on the reefs that sponges once were, and now there's shifts back and forth? Is there any fossil records to indicate, or any data to show something of that nature?
So Andrew's question is, the Earth has undergone several catastrophic biodiversity losses in the past. The most recent one, of course, you're all familiar with. It's the Chicxulub impactor event of 62 million years ago when the dinosaurs went extinct, right? Since then, we haven't had any major extinction events except the one that's going on right now, which is the Anthropocene extinction event, and it started about 10,000 years ago, or some people say 30,000 years ago, when the major megafauna on land were killed off by us. So your question is given that there have been other times in the history of life on Earth when there have been dominant groups of organisms that have sort of taken over and then sort of lost their prominence because of an extinction event or whatever, he asks, does it look like maybe sponges might be, ascendant while other groups may be not ascendant? Interestingly, there's one group that studies sponges in New Zealand that suggests that we are moving towards sponge reefs, not coral reefs, but sponge reefs. So that is, in fact, one of the ideas that are out there. That in the Anthropocene, which is the current extinction event that we're in, corals will largely go extinct. We will lose the ability of corals to build reefs, and instead all the hard benthic habitats in tropical zones will be dominated by sponges.
Do they fossilize well at all to show?
They unfortunately do not. There was a time in the Paleozoic when sponges had, and they still exist actually. They're a living fossil called sclerosponges. If you ever dive in caves in the Caribbean, which it's not entirely recommended, but there are these globular sponges that secrete incredibly hard endoskeletons that grow incredibly slowly, and they're still around, but there was a time when they were dominant, and they left a wonderful fossil record. But most of the sponges out there fall apart when they die, and the most you can look for is their spicules actually in the interstices of the reef when you do coral boring. Interestingly, since our vicious circle hypothesis has come out, there was a group in Panama that bored down into the reef and looked at the sponge spicules, and it turns out that the sponge spicules of the palatable sponges are different than the sponge spicules of the defendant sponges. And what this group discovered was that the spicules change exactly at the timing that we had in our prediction based on when hawksbill turtles were decimated. So you see the change in the spicule record that is reflective of the loss of hawksbills in Panama. And for Panama, it's even more interesting because the Aboriginal people had started fishing out the spongivorous fishes first, but they hadn't actually gotten to all the turtles. Then the Europeans came in and wiped the turtles out. So there's this sudden shift in the spicule record based on that. So that's the first corroboration of the vicious circle hypothesis from a historical geology study.
This will be an easier question.
All right.
So if the European explorers wiped out the turtles in the Caribbean, were they not doing the same thing in the Indo-Pacific?
They weren't, because the Indo-Pacific is too big. They really had a smaller pond to work in, and most of the Indo-Pacific, in particular the Coral Triangle, was just it was already had Aboriginals there at low population levels. I'm talking about Borneo, Indonesia. We've really only started to ruin that area recently. It was in pretty good shape right up until the 50s and 60s, and it's gone downhill amazingly fast. And it's just because the population levels there were very low, and Europeans didn't want anything from there. Anything they wanted, they could get in abundance from sort of cities, so spices and that sort of thing was easy enough for them to get. The Caribbean is smaller, and so the impact was greater.
The sea urchins come into your circle of...
Well, the sea urchins are good question. Where the sea urchins come into play? Well, certainly their loss was a dramatic importance. The 1-2 punch of losing corals and sea urchins at the same time in the 1980s from different pathogens was devastating for the Caribbean. Sea urchins seem to be coming back in very specific places. And it's not clear whether it's because they are resistant to the pathogen or whether they're just like in places where the pathogen isn't active and then the pathogen moves back in. But there is some indication that they may be coming back. They eat algae, just seaweed. So, and in the places where there are lots of them, the reef looks great because the coral and algae comes back too. And the coral and algae is the substratum that the corals recruit to. So the babies land there and grow into corals on the pink coral and algae. So the more sea urchins we have, the better. In fact, if I were to push one reef restoration technique above any others, it would be for people to put down racks of PVC where the openings of the PVC piping is up because the baby urchins recruit into the openings of the PVC and they can survive all of the fish that eat the babies. That's their biggest problem. So the sea urchins like to live under the spines of the adults. If you don't have adults, the babies get eaten really quickly. I mean, it's amazing. If you take them out, especially the black durgans, the triggerfish will have them in a second. They just eat them up. They love them. So they will go into those PVC pipes and spend the day and come out at night and graze around and get big. And once they've reached a certain size, the fish can't get them anymore. Despite that, they still hide during the day, even when they're large. So forget about coral restoration. I hope I didn't offend anyone here, but yeah, that's the key. Get the diadema, because it's amazing. You grow up the corals. They do great when they're on lines. You put them on the bottom, dead. It's so sad. It's just tragic. It's like all that work. And I can't imagine what it's like for these people that spend years raising all these corals. They put them on the ground, they go away, they come back, and they're all dead. It's like, and they don't like to talk about it, of course.
Designing this ant creation that you put in the garden to repel ants, the opposite, to attract like larvae and protect them, and also the right chemical. But that's the next 4 years, I don't say, but actually going to get happen. So
Yeah, very often the things that you grow in these ways, they become like hothouse flowers. You know this expression? They can only survive when you're keeping them suspended above the reef. The minute you put them down, they can't take it. They're not used to it. So better that you should get the babies there because they actually grow up in that environment and they do better because of it. But it's the same thing. Similarly, people have raised sea urchins. It turns out it's easy to raise them in the lab. You can raise lots of them. And the first time they did that, they came out with all these baby sea urchins. They put them on the reef, and the fish came and all gone. Yeah, it was like all this work, and the people that were doing it were just like. So better you let just nature do it. Question in the back? Oh no. Okay.
Do you expect, with all the rivers going into the Caribbean Sea, do you expect a lot of effect, and if so, when maybe, of all the fires at the moment in the Amazon and the Orinoco and things like that?
That's a wonderful question. So she asked whether, given the current state of the Amazon and the fires going on there, whether that might have an additional impact on Caribbean reefs. I have to, you know, the bit about the DOC from fresh water is more speculative than anything in this story. And the reason it's more speculative is because the interesting thing about dissolved organic carbon is we don't know what it is. We can measure it in total, but it's very hard for us to identify the individual molecules that are in this mixture. We know there's this spectrum of labile to refractory, so everything from sugar that we could eat and taste sweet to the tannin, the brown color in your coffee, that's all present. But our ability to identify those compounds is extremely limited. So we hypothesize that there may be differences in the amount across the spectrum and the types of molecules that are present, and that mixture has changed over time. But to answer your question, it's entirely possible that by removing a lot of the plants that have existed for eons, that have been involved in that ecosystem, that there may be fundamental changes in the kind of molecules that wind up in those rivers, that wind up in the surface water of the Caribbean, and that might have unforeseen effects on the sponges, either positive or negative. So yes, it's entirely possible. We're just now working on identifying the molecules that are in dissolved organic carbon. In fact, our most recent proposal to the National Science Foundation is just about that. So,
the big picture, trying to understand the worth of sponges. As you were talking about, I'm thinking, they're cleaning up as they take this water and they're cleaning up, getting rid of sugars. But then you say they're peeing. How valuable are sponges locally, regionally to the ocean? I mean, since they're peeing a lot,
right.
I mean, what is the value to our water?
So the question is, what is the value of sponges given that they have both obviously positive and negative effects on the water around them? They're cleaning the water, but they're also peeing in the water, right? Well, I would argue that there are some real negative consequences of sponges. They're taking over territory. They're basically involved in killing the remaining corals, right? From the standpoint of the dive community, they're probably the prettiest thing on the reef. So, and basically it's all we have left. So we could have the algae, which are not very appealing, or the sponges, which are. They have interesting morphologies, beautiful colors, and they're actually actively removing particles from the water, so they enhance the visibility. So they definitely feed the spongivorous fishes and turtles, so they're a food source. They're important in the ecology.
What's the quality of the water?
Well, you know, if you're talking about removing particles, that's a positive. If you're talking about the fertilizer, it's a negative. So it's complicated, right? But, you know, what I like to tell the people in Florida, which are desperate about the lack of coral, now the most recent problem has been this thing called, stony coral soft tissue disease or whatever. It has an acronym. Florida's corals are doomed. And to a degree that's so much worse than even it was 5 years ago. What I like to tell them is you have beautiful sponges. You know? Promote your tourism. Go look at the sponges. So what's the negative consequence of that? Well, the reefs are being built by the corals. In their absence, there will be no reef building. And what that means is the rocks flatten out. You're going to get sand and lower topography, which means waves and energy is going to make it to the shore, right? That's not an issue here in Ceiba. You're on a volcano. You come out of deep water right to the shoreline. No worries. But in places where, like the Mesoamerican reef along Belize, Yucatan, Mexico, Honduras, that thing is going to be flattening out over time, which means more energy is going to make it all the way to shore, which means, you know, in addition to global sea level rises and stronger hurricanes and higher surges from stronger hurricanes, all that energy is going to make it to land and wash right over.
Maybe
Well,
you
we're
know,
going to lose a lot
species.
more people. Yeah. The fish have ways of dealing with storms, but
Species will die without reefs.
oh, species are dying. Yeah, regardless. Yeah. So, other questions? Great.
Not that this has ever happened to me, but hypothetically, if a diver were to touch a sponge, afterwards, there might be a slimy sensation on your fingers.
Oh, good question,
I have no
Sabrina.
idea what this would be like, but if if one were to imagine it, what is that?
Okay. There are several sponge species in the Caribbean that have extremely nasty dermatitis-inducing compounds, which are sugars, interestingly, but they're surface sugars. And the one that's common here is the touch-me-not sponge, which is called Neophibularia nolitangere. And it's brown. And it's brown and forms like little barrels that are connected to one another. They're notable for having lots of little worms that live in their tissue. And if you touch that sponge, you are in for a world of pain. A very specific kind of pain. So I'm assuming most of you have dove and you've probably been hit by fire coral before. That's its own distinct kind of pain, right? The way it feels when you're originally stung, and then the feeling that you get afterwards. It's like you get this cold, clammy sensation. You all know what See, it's so interesting for me because back in the 80s and 90s, everyone touched everything underwater, and now no one touches anything. So when I talk about how different stings are, people just look at me like I'm crazy because basically we used to touch everything. So, and the worst of it was what would happen. We used to get permits to collect sponges to determine what secondary metabolites, what chemical compounds were found in their tissues, and we would provide those compounds to drug companies for testing. And so we would collect large amounts of sponges, and we would bring them back in bags, and we would get them on ourselves in the process of collecting them, right? So we touched them a lot. But worse, very often those bags would spill out in the boat, and we'd get our feet in them. And there was nothing worse than if somebody spilled the touch-me-not sponge and that liquid, just the liquid, in the bottom of the boat. If you were wearing sandals or even booties, oh my God, for 2 days, this intense, very bizarre cross between pain and itch. It's nothing like it. There's another sponge that's found in the mangroves and usually on the bottoms of boats or floats called fire sponge, bright red, Tedania ignis. Once you've seen it, it's really obvious, bright orangey red. You touch that, nasty dermatitis, very different than the touch-me-not sponge. Similarly lasts several days. For me, if I'm anywhere close to it, my fingers blow up like little balloons. Really weird. Yeah. So.
That I would never have experienced.
The slimy feeling is also species dependent. It's mucus. Yeah, so same as, but there's one sponge that if you cut it and pull it apart, the mucus is so elastic that if you release one of the halves, it snaps back together. Yeah, it's like elastic bands, yeah. So, and again, this is something people don't experience because no one's allowed to touch anything, which is a good thing, right? There's also one that's very common here that if you cut it, it will bleed a brilliant purple. And the color of the sponge changes from yellow to purple as you watch. So, very, very neat. Yeah. It was really fun. Yeah, God, the things we used to do. Oh. Other questions? All right, well, I want to thank Lynn and the crew for inviting me. It's one of I like to tell my colleagues I love this because it makes me feel like I'm a rock star of some sort. So this is a lot of fun for me to come here. So thanks for coming tonight. Yeah. Oh, right. Sorry.
Just have a
Oh,
quick.
yeah. Okay, you got the
Got
clicker.
it.
Okay,
All right.
great. And I will sit down.
Can we have another round of applause for Joe for his very fascinating talk on sponges? And thank you all of you, both live and Facebook, for joining with us tonight. We encourage you to stick around for drinks or some food afterwards. We'd like to thank all of our sponsors. And then we have a few more upcoming events in the next week. Tomorrow, Joe has his field project where you can meet us at Fort Bay Harbor, or you can also meet us at the tent for a ride down in a taxi. Then again on Monday, we have our next presentation by our seagrass expert, Anna Mates, at Brigadoon. Tuesday is her field project, which you can also meet us at the tent or at Fort Bay Harbor at 1 o'clock p.m. to collect and analyze some seagrass. And then another presentation Wednesday night at Long Haul by her husband, Eric Bowman, who is a Queen Conch expert. Thank you, and please join us afterwards.
Thank you. I'm joking.
Do you want to come up?
Anybody that's not going snorkeling or diving tomorrow, there's going to be a drop-in class at my studio at 2 o'clock, and for volunteers, it's free.
Miami is known as an international community, a hub of trade and logistics for the Caribbean and America. A leader in the freight logistics industry, Caribtrans has served the Caribbean region for more than 30 years, with a footprint that has grown from Long Island to now serving the majority, as well as Central and South America. Caribtrans is an NVOCC, a non-vessel operating common carrier, meaning it has the same responsibility as a shipper, vessels or planes. Its primary customers, individuals who ship clothing, electronics and other personal items, and occasionally cars.
Most of the stuff that they're sending is because they get there. So for us to provide it on two services, air and ocean, we can give it a choice of how fast you want it. So if you want it really fast, we're going to send it by air, you're going to have it next day, sometimes even the same day.
That on-time service has been the key to Caribtrans' success, whether shipping by air or by sea. The company moves about 5,000 TEUs of freight each year, more than any NVOCC in the region. And since it joined the Pawlik family of transportation companies a couple of years ago, Caribtrans can offer customers even more options.
Other NVOCCs, they depend on other lines that are not part of their family. So they need to rely on whether they're going to sell or not, whether they are late or not. It doesn't happen to us because it's part of our family of companies. We control the service that we give.
Sí, buenos días, por favor, con Diana.
Quality service is what keeps customers coming back. Customers like family, and when they leave to research competitors, it doesn't usually last long.
They have left and they have come back because they've said that, nobody does it better than Caribtrans. So because of that relationship.
That exceptional service includes consolidation. Customers can have freight sent here and stored for free for up to a month, then consolidated and shipped on to the islands and Americas, a perk for the region's growing small business market. Puerto Rico is an expanding market for Caribtrans, about the size of Connecticut and rich in history and culture. Puerto Rico is home to many pharmaceutical manufacturers, as well as a thriving tourism industry for which Caribtrans moves raw materials and supplies. But serving small businesses is still the company's bread and butter here in San Juan. Businesses like Caldera Café, a local coffee company. Owner Alfredo Rodriguez started growing coffee beans at his mountain farm about 20 years ago. He recently ordered a variety of equipment for his farm and shop. He used Caribtrans because it consolidates, cuts down on the number of times he has to pay the Puerto Rico import tax.
companies want to do that. They want only big cargos and maybe half container or full containers, but they don't want to consolidate because it's more difficult.
Opportunities is what Carib Trans does, and includes helping to grow small businesses like Caldera Cafe by taking the hassle out of shipping.
We can be a gateway to expand their business and the U.S. Wherever they can sell their products, we are also to be a partner for them.
That customer dedication has made Carib Trans what it is today. Now, as part of a larger family with more transportation resources, Carib Trans hopes to grow beyond its current market, offering services to additional Latin American ports and eventually Europe and Asia. But even when it becomes a global logistics company, Carib Trans wants to stay true to its roots, always providing personalized, reliable customer service and a commitment to the communities it serves.